In this lecture, I'd like to provide a little overview and specifically focus more on RNA processing, but we'll start at the beginning with DNA. So we start with DNA that's transcribed into messenger RNA. The messenger RNA is then translated into protein. So typically we go from the 5' end to the 3' end. So the transcription happens opposite end, right? So if we start on the 3', we're going to transcribe out the 5' end comes out first, and then the 3' end ends it. 5 to 3, and then N to C. So the nitrogen side, the amine group, is going to come out and associate with the 5' end, and the 3' end is going to be on the carboxylic acid end. This should again be reviewed, but we have different G, C, A, and then uracil or thymine, or uracil and thymine. Those are the differences in RNA versus the DNA. Certainly also an included difference is that backbone carbon, the sugar backbone. If we have the deoxy, means that the hydroxide part is removed. So the deoxy, that's what the change is between DNA and RNA, besides the uracil and thymine base. Sometimes people say that DNA is double-stranded and RNA is single-stranded. It's kind of true. There's two different separate strands. There's a 5' and a 3' end for both, but RNA can double bond, hydrogen bond. So you can see that here for a transfer RNA, for example, here we have the T loop. We have these different red spots showing us how the G's and C's and A's and U's from one single strand can still hydrogen bond and pair up. These structural features and abilities are really actually important for the how this is able to be recognized by other proteins and how it's used for this specific area, for example, for the anticodon loop, and how it's interacting in the cell, and helps to preserve its ability to not get chopped up and broken down in the cell. Also, amino acid attachment sites, there are signals on here that inform the cell where this is supposed to shuttle to, what it's supposed to do. So that structure and the function are all combined as well, just like in a protein. Of course, the DNA is in the nucleus. It's transcribed in the nucleus. Different splicing, capping, polyadenylation, that all happens in the nucleus as well. And it's not until splicing, capping, and polyadenylation happens that it's able to be exported out of the nucleus and into the cytoplasmic area where it can then be translated. So all of these things have to happen. This processing in eukaryotes must happen before it's transported into the cytoplasm and ready for synthesis if it's an mRNA. As a general rule, we can have different amounts of RNA. So if I have gene A is transcribed and I get a lot of RNA, that typically holds that will produce more protein. And if I have less RNA produced, that makes less protein. So a lot of the times we'll use RNA amounts to inform us how much protein we think we have. However, I just want to mention that there are other processes and other things that can happen. Protein might be modified, or the RNA might be modified or changed or held in a place where it's not translated. So this isn't always the case. So generally, yes, this is true, but there are situations where this 1-to-1 ratio, for example, does not hold. Typically the amounts of RNA, so if we're looking at this slide, so how much, if we want to kind of get an idea of how much protein might be synthesized or changes, we want to look at how much RNA there is. And usually we're using PCR. And the Q here is for quantitative polymerase chain reaction. So quantitative means we're getting numbers out of it. So how much of it are we getting? This simple process, we look first, if we are starting with RNA, we have to reverse transcribe it from RNA to DNA, because PCR really only works with double-stranded pieces of DNA. So we have to make a cDNA library. And so we start with a primer set. Sometimes a poly-T is used as a primer. If we have a poly-A tail, we can use the T to prime it, and it can read backwards. Or we can use some different hexamers that are randomized that will attach to anything. But it's important that we make this other strand, and then from that we can heat this up and denature, and then we can anneal our primers onto whatever gene of interest we're looking to overexpress, and then we can elongate through polymerization, and then we repeat the process over and over and over again. Usually this is done in the presence of this cyber green. So as this is elongating, it's going to incorporate different types of fluorescent molecules in the DNA sequence. And using that, once it gets incorporated, you can see here that cyber green is off. Once it's incorporated into the DNA, changes its structure and it's activated, and then it fluoresces this green color. So then we can look at the amount of green glowing in our experiments, and then that can give us an idea. We can use that to figure out how much of our RNA we started with at the beginning. So again, this is called quantitative PCR, polymerase chain reaction. And so what's going to happen as we're elongating and doing this over and over and over again, the cyber green gets incorporated, and then it reaches this certain threshold value that, okay, so now we know we have this. And typically the threshold, depending on how much we started with, how much template we started with, that threshold value is going to change. If we have more, that threshold is going to be less cycles of the PCR reaction. And if there's less of our starting point transcript, it's going to take more cycles to see that. So you can see that in this next figure. If we look at the threshold value here for this red versus blue, green, so there is more of this copies, either 10 million copies of whatever the template is that we're looking at here versus a million versus 100,000, right? So the less and less we have, the threshold value will change. So the cycle number changes here. So here's some data from a qPCR run. Each line represents a different gene. According to this run, which gene is most highly expressed, and I just mentioned that would be this red line here, because we have the most of that template. It's going to take less cycles to meet that threshold value. Typically experiments that are looking at the threshold value, they'll report that. So I want you to report the threshold value for all of these different colors. So take a little bit of time, pause, look at all of these 1, 2, 3, 4, 5, 6, 7 different colors. What is the threshold value that would be reported perhaps in a transcript or a manuscript for a paper? So an example of this would be the threshold value for red is probably maybe like 14 or something like that. Again, here's some more information as it might be shown in a figure. So I'd like you to pause this and read the above text, and then I'll talk about this in a moment. So first thing I'd like to point out, this fold expressions of mRNA. This is where we're looking at that the threshold value. Essentially, it's changed a little bit. We're looking at different numbers. So they've converted the threshold value into an absolute amount of mRNA, but it's a similar kind of idea and concept. In the wild type, they've normalized those levels so that whatever normal expression is, they've normalized it to a 1 in that instance. This PLS3 is a transgenic construct, so it's been genetically modified, and we see changes in the levels of these gene transcripts in these different tissues. The first question, what do the results below tell you about the reporter gene's expression? Where does it get expressed? Well, we see that it's at a 1 in all of these tissues. So we say it's expressed everywhere that it was tested. Of course, we were just talking about quantitative PCR, so this is how we would generate these data. And the overall technique, again, we're looking at messenger RNA, but in order to do qPCR, we have to convert it from mRNA to a cDNA library. So we would start by getting our cells, isolating the cells from the brain, spinal muscle, liver, all of these cell tissues. Then we'd lyse the cells, we'd break them open, purify the messenger RNA, reverse transcribe to cDNA library, and then use the PCR primers for the gene of interest to do a quantitative PCR. So back to these big picture idea, we start with DNA. We're going to synthesize, we're going to transcribe that from 5' end, so it's going to use the template strands starting the 3' end to 5'. It's going to be anti-codon. We transcribe that from 5 to 3 and make the transcript out. This here is shown in this figure where we have the RNA polymerase taking this template strand. It's going to add the new transcript here, 5'. This 5' is going to be the N-terminal side of our protein as well. So these are coming in and getting added onto the growing strand. We can see here that multiple RNA molecules, we have this mRNA coming off of here, off of the DNA chain. These can happen simultaneously. So this can be kind of like a train synthesizing new mRNA as we go. So remember when we opened up the chromatin and this becomes available, this really helps to recruit some of these, the RNA polymerase to kind of synthesize these new strands. However, mRNA is not the only thing that's transcribed. So that encodes for proteins, the ribosomal, microRNAs, transfer RNAs, small nucleolar, small interfering. There are all sorts of different types of RNAs that regulate different types of processes. So these are all important, and perhaps we'll have a chance to see some of these in some of the presentations that are happening in the last week of class. I'm not going to spend too much time on this, but there are sites that are encoded within the DNA that help to bring in proteins that recognize starting and stopping points for this. Again, this is more focused on in other classes where we have promoters and terminator sites as well. But they're encoded within the DNA, and those areas are then recognized by proteins that help to modify that function, help to bring in the polymerase, and then help to drop off the polymerase as well. One thing that's interesting is those different promoter regions actually recruit different polymerases. So if you have, for example, a promoter region that recruits RNA polymerase 1, those are mostly ribosomal RNA genes. Polymerase 2 is going to be recruited in areas where we're going to generate protein coding, so mRNAs, and then microRNAs, so things that also have different functions as well. And then Pol3 is going to be recruited areas where tRNAs are going to be expressed, ribosomal RNAs, so other small RNAs that have other functions that are not necessarily gene encoding. So it's interesting, there are 3 different polymerases. Those DNA sequences recruit these different things that are going to have different functions. It's interesting how that's split apart. I'm not going to spend a ton of time on this, and don't memorize this for anything. I'm not going to have it on any exams, but just to give you an introduction to this idea. There are a lot of, again, these proteins are recruited to the double-stranded RNA, and that helps to bring in and recruit different polymerases. So an example, Pol2 here we're looking at because we're interested in looking at mRNA expression in this example. So a lot of these different components are going to be brought in and recruited to help to start the mRNA gene expression. So that the TBP, which is the TATA binding protein, it's actually going to bind to that piece of green here that is that TATA sequence, and actually helps to bend that. So that's where it kind of opens this up for something to come in and to kind of break apart these hydrogen bonds, that base pairing activity, so they can get in there and they can synthesize just that one template strand. One of the first initial and essential steps the RNA polymerase 2 does is that actually has this little tail here that's phosphorylated, has capping factors that are bonded to it, splicing factors, polyadenylation factors. So as this chain grows out here, it is able to interact with all of these, but it must first interact with the capping factor. And the capping factor will bind to the growing 5' end of the mRNA, and it adds this different type of cap. And what does that look like? Here we have a normal, these different bases of the backbone sugars, and the bases are here. The 5' end of the cap is actually, this is the normal way that it would be, but then here's the triphospho bond here, and the cap is actually flipped around, so it's not going to be the same sequence and orientation here. Actually reverses it, and it helps to identify the 5' end of the growing mRNA. So this is an essential, this has to happen for the mRNA to get exported out of the nucleus. So here you can see the cap is going to happen. Then we have different coding and non-coding sequences, and then the polyadenylation. So we have to have cap, polyadenylation, and then we're also doing splicing that has to get out of for the mRNA to get out of the nucleus. After capping, the next processing event that happens is a splicing event. Those for all eukaryotic genes are going to be spliced in order, and splicing factors have to be in that final gene to get exported. So we have different regions that are exons, they're coding regions. These are what are going to be encoding the amino acid sequences that will get translated. And then the introns, those are non-coding regions. So they're regions that are not part of the final gene profile or the protein that's going to be synthesized. So I will just mention that homologous recombination is something that happens during DNA replication, and different parts of those are involved in DNA. But it's an analogous idea where there are different sections and components. So different types of transcripts can be synthesized and different exons can be cut out. So you can maybe lose this part and just put these and these together, or this one and this one, or this one, and that last one. So there are different ways to take these genes and splice them differently and get different outcomes. So it's kind of an interesting way that you can get some variability from the mRNA as well. Just like on DNA, there are also sequences that are built into RNA that help to for the cells and different proteins to recognize where things should be spliced. So these things that recognize these splice sites are typically small nuclear ribonuclear proteins. So they are part RNA and then also part protein. So there's a combination. So those are called snRNPs. So these are the sequences that are recognized to help identify the gaps or the parts that are going to be cut out. So the AGGU, RAGU, you might wonder what R means. It is either, an R is either an adenine or a guanine, a Y is a cytosine or a uracil. So these are going to be the sequences that are recognized. So there's one on both ends and then one in the middle actually. So these are required for the intron to be removed from the mRNA. So I mentioned the small nuclear ribonuclear proteins. So ribonuclear means we have some RNA that is also synthesized, typically by like a Pol-3 polymerase, and it's associated with a protein that's kind of shown in green here. So the RNA helps to double strand bind to that sequence, recognize that sequence, and then the protein here. These proteins then come together with another binding site, a binding protein in the middle that's going to recognize that intronic region. And so that comes together, and these are cut off. It turns into what's called a lariat, and they're cut off and removed, and this is recycled and used in other parts of gene expression. So these come off, and then we have exon 1 and 2 now spliced together. So I mentioned before there are things called alternative splicing. So we have the pre-mRNA splicing. So here's the pre-mRNA, and then we have different splicing sites. So we have exon 1, 2, and 3. We can have just 2 and 3. Maybe we just have 1 and 2. So these create 3 different mRNAs and theoretically 3 different proteins. After the capping and the splicing is happening, as these are kind of growing this chain is growing, the final event is the polyadenylation. So we have a cleavage stimulation factor and a cleavage polyadenylation specificity factor. And those are going to be recognizing an A2UA3 sequence on the back end. They're going to bind, they're going to cut that off and polyadenylate it. So here's another, just showing you that binding, that group of proteins that are binding to multiple different regions, sequences within the messenger RNA, and they are going to cleave and cut this off here. And then the next step is to recruit different factors to the end here and polyadenylate it. So here then, once we have this group is still bonded, we have these factors recruit what's called a poly A polymerase. poly binding protein, and then we add that longer tail on the back end. So that event is necessary then. So we, again, as we're growing off of, we have our DNA, we have the RNA as it emerges, it's going to get capped, it's going to get spliced, it's going to get polyadenylated here, and then it's allowed to be transported from the nucleus to the cytosol. All of these processes change what's happening and change kind of the flavor of the message. So we have the cap binding proteins. There are proteins that are going to be associated with that 5' end. We have proteins that will be associated with that exon junction, and we'll have poly A binding proteins. And there are different pore complexes that are going to be looking for all three of these things before it's allowed through. And if it doesn't have these three things, then it doesn't get through, it doesn't get to the cytosol, it doesn't get translated. So that's how those things, all of those processing events kind of leave its mark on this to let everything know that it's ready to go on to the next stage. The cap and the associated proteins, as well as the poly A tail and associated proteins, actually help to then recruit, once in the cytosol, the translational machinery. So it helps to recruit the ribosome. So a big difference between eukaryotes and prokaryotes is that eukaryotes have a nucleus. So all of these events are part of that processing, and they're required before translation. Prokaryotes don't need that. They just have mRNA. They don't have capping, polyadenylation, splicing doesn't happen. What you get is what you end up with pretty much in terms of prokaryotes. But just recognize there is a difference, and these differences are really important to understand depending on what system you're studying this in. So once out of the nucleus into the cytosol interacts, it gets recruited by a ribosome, and there are these different codons that are going to signal for different amino acids, the 20 essential amino acids that we have, and then also some stop codons that help the cell know when to stop making the amino acid. I like this one the best, where we're looking at the 5' end. So GGA is a glycine, GUU is a valine, ACC is a threonine. Kind of fun to look through this. I think I like this one the best. It's kind of the most organized. It tells us a little bit about the type of amino acid that we're putting on, if it's basic, acidic, polar, nonpolar, or lipophilic, or whatever you want to talk about there. So that's kind of interesting how this is set up. What's really interesting is that you'll notice that some mutations actually won't matter. So if I go G or AGG and I mutate that last G to an A, so AGA, that's still going to give me arginine. So even though there might be changes in what that overall outcome is, it's not always going to have an influence on the final protein product. All right, so I'd like you to practice this to kind of get what would this set of, what would this be, what would this give us for, and if we had an mRNA that looked like this, what amino acids would be expressed from this sequencing? And I'll just give you a start. If we did C, U, C, we're going to have a leucine that's going to be the first one. So practice the other few on here, and let me know if you have any questions or problems with that. Here's kind of another learning check in terms of your ability to kind of walk through this. And this is a segment of DNA from a transcribed region of the chromosome. So I would like you to kind of look through this, make sure that you can answer these two questions. And again, I'm not going to give you the answer, but if you feel like you're not sure of the answer, you would like to know what the answer is, we can talk about that separately. But this is something that you should be able to do. This is just to demonstrate reading frame matters. So where we start that reading of 3 is very important. It'll give you different amino acids. So that's why the methionine start site is typically important to know where that is so that you can be in the right frame. Of course, as we're adding those amino acids to the chain, there again, an important component of that is a transfer RNA, and it does have a 3-dimensional structure with important components and loops and areas that help to read the anticodon region. They're associated. This region is associated with an amino acid that's attached that's going to build onto the growing amino acid. So just to kind of learning check here, if this is the anticodon region, 5 prime, 3 prime end. We know we bind to those in an anti orientation. So what is the amino acid would be attached to this transfer RNA with the anticodon shown below? So think about that, work through that, pause right now, and I'll talk about that in a moment. Okay, so the bottom part, this is the part that is going to be important for what is going to be on attaching to that. So that's the component that's going to dictate what is the transfer RNA going to be. We know we're going to look at for the 5 prime end, this is the 3 prime end, and this is the 5 prime end here because it's going to be an anti-sense orientation. G is going to match up with a C. A's are going to be matching up with U's. So it's going to be CUU, except for this is the 5 prime end. So we need to start with that end. So if we look at U, C, phenylalanine is going to be the transfer RNA, the amino acid attached to this transfer RNA. Here's another one to try out. I'm not going to give you the answer, but if you feel like you have a question about this, please let me know. So these amino acids are attached to the transfer RNAs, and then these are used in translation, as we've been talking about. And the ribosome has different exit sites, a peptide growing site, P site, and then also an amino site. So these are the different regions where they'll bind, the transfer RNA will bind, it'll transfer and make the peptide, and then that transfer RNA will exit. So this initiation factor binds this along with the initiating transfer RNA, which is always methionine, is going to find and scan for that UAG site to bind to it. That'll bring in the larger ribosomal subunit. And then at that point, this chain starts to grow. So the new charged transfer RNA will bind to this region within the ribosome, and then this all shifts over, and you can kind of see the different sites as it shifts over here. Then this top part moves over, and then that synthesis occurs, that peptidyl bond gets made, and then 3 gets bumped out, and then this kind of moves back over here, and we're starting with, now we can have an amino acid come back in here and ready to go for another round. A lot of antibiotics are actually inhibitors of protein synthesis, so it's something that will inhibit bacterial proteins or RNA synthesis sometimes, but that's not typically our ribosomes, and our polymerases are kind of different enough from bacteria where they don't tend to have interfering or issues within people, at least in low doses. So tetracycline, streptomycin, chloramphenicol, cyclohexamide, rifamycin, those are all different things that are typically binding areas where transfer RNA will bind to, or it's messing around with the RNA polymerase. So these are kind of, most of our antibiotics are based off of binding to pockets where these processes are occurring. And this slide is just kind of remind you that RNAs don't only encode for mRNAs that encode for proteins. So the ribosome is made up of huge amounts of RNA. It's really a ribozyme, and it suggests that our first ancestral cells actually utilized RNA, not proteins, as enzymes, as things that did things within our cells. So rather than enzymes, we're talking about RNA-based enzymes. They're called ribozymes. There are a lot of different things. We can form peptides, DNA ligation, splicing, polymerization, phosphorylation, alkylation, bond rotation. So they have a lot of different functions, and they're based off of RNA structures. An interesting thing about ribosomes, a lot of times they'll actually probably more, obviously it looks almost more like a snail shell or something, but these might form more of like a ring or a circle where these polyribosomes will bind. They'll be making protein off of this transcript, and then they'll just hop back on. They'll get recruited back by the 5' cap after it's done, and then it'll continue the cycle. So these many actually will jump, and then this will just get processed until eventually it gets broken down and degraded. And we spoke about ubiquitination and bringing things into the cell targeted. There are things that a lot of protein breakdown really helps regulate the amount of protein in a cell, as well as the amount of transcripts. So when we want to get rid of proteins, they can get modified, for example, by ubiquitin. That's recognized by other proteins that help break down and degrade the protein into the amino acids that can be recycled and turned into different and other proteins. So that really is the overall outline for the lecture. It's a lot of this should be review, but I wanted to make sure that we covered some of this in case it was a little fuzzy from other classes as we move into looking at some of the regulation of RNA and some of these processing events that occur that help us understand the how RNA is important for cell function as well.